Hey guys, welcome to the other part of chapter 12. And in topic 2, we're going to talk about ion channels, and then we're going to tie together ion channels and transporters to discuss in depth how nerve cell signals work. So as I said, we're going to talk about ion channels in this topic as well as nerve cells and how they tie into membrane transport. Here are your objectives for this chapter, or for this topic rather. Make sure, some of them overlap a little bit with the last topic, but it's important to understand how this topic and the last topic compare to each other and how they work together. So let's first talk about ion channels. We're going to talk about what the properties of ion channels are and how they're controlled. So it's really easy to think of an ion channel as a pore. And ion channels are not pores because they are selective. They can they're selective based on size and shape of ions, or even on charge. So they are very selective. They're not just an aquaporin that allows water to flow in and out. They actually have a lot of control over what's happening. And they can be open and closed as needed. Now granted, unlike transporters, which have a little bit more control over how much is transported, ion channels, when they're open, just allow for massive influx or efflux of the item that it allows to flow through. So it's more of an open versus on-off switch versus medium heavy light that we see in the transporters. But as you can see, they have this barrel shape with a hole in the middle, and there's proteins on the inside of the barrel that allow for that selectivity we were talking about. So how are they controlled? There's three main types of controls in ion channels. There's voltage gated. We're going to talk a lot about voltage gate and how this works here in a second in nerve cells. But basically, the electrochemical gradient is what helps open and close them. Then we have ligand gated, which is something where a ligand has to bind. We'll change the conformation of the ion channel and allow it to open or close. And then we have stress gated, which is actually where there's a physical change, a physical event occurs on that gate that allows for it to change, for it to open or close. So before we get into our examples here of how this works, we got to talk about what a nerve cell is. Some of us maybe haven't talked about nerve cells in a while. Nerve cells have quite a unique structure. They have the cell body, which is the main area that we've always talked about, with a nucleus in the middle, and you can see an example of that here. And they have these long spindly sections coming out called dendrites, and these are responsible for receiving signals. Then they have what's known as an axon, which is the very long arm that spreads down to the nerve terminal, which is what releases the signal to the next cell. And the axon is responsible for carrying the signal away from the cell. So make sure you understand how that works, because it's going to play a role here in just a second. So let's talk about how nerve cells conduct signals. Now, we've talked a lot about action potential and membrane potential. And action potential is very similar, but action potential is different because it's self-renewing versus membrane potential, which is not. So let's look at an example of how this action potential is generated using sodium channels. So what happens is we have this sodium channel, and it's closed. Then it becomes open, which changes the membrane potential of the cell. And you can see that here on the far left, where the curve is starting to go up. As the curve goes up, eventually it's going to reach a place where the channel becomes inactivated. Now, if the channel didn't become inactivated, there would just be a massive influx of sodium into the cell, and it could cause all sorts of havoc. So the channel has to become inactivated, and it's triggered at this point to be inactivated about a 50 millivolts. When it's inactivated, it doesn't matter what's going on, it's not going to reopen. And this allows for the membrane potential to then peak and go back down. And after it reaches a depolarization area, or space, time with the millivolts, then the channel will reset into the closed conformation, waiting for the next signal. Now, how this works in the cell is what it does is it's going to stimulate down the chain of the axon, or throughout the cell, depending on what we're doing. And this is called propagation. So what happens is the one will be stimulated, then the next one will be stimulated, then the next one will be stimulated. And as this flow is going down the axon, the ones behind it are becoming inactivated, then eventually will be closed, and then eventually can open up again to send the signal again. So that's how this action potential works. And I've put some videos up in Blackboard, so if you have any questions about it, check that out. It's a little bit easier to see. So let's talk about what this means in terms of the cell, because we're not worried too much. I mean, I want you to understand how nerve cells work, but the big thing I want you to understand is how this works with relation to cell communication and everything else. So you can see this here where we convert electrical signals to chemical signals. So what happens is that signal is sent down the way, and eventually something's going to happen that's going to trigger a response at the end of the cell. And you can see this here in a calcium example. And as I said, calcium is hugely important in the cells. You can't underestimate how important calcium is. So in this example, the calcium gated channels, and they're voltage gated, the voltage gated calcium channels are closed. They become open through the signals that are sent from the rest of the cell. This release of calcium into the cell is going to release the synaptic vesicle, which contains neurotransmitters. And these neurotransmitters are going to then be released across the synaptic cleft to the next cell. That is then going to either use this to change the membrane potential, which is the electrical signal within the cell. It's going to create a cascade of events depending on what that cell is. It's all about what that target cell is that determines how these neurotransmitters are going to work. Acetylcholine is a neurotransmitter receiving receptor on brain cells and other cells. And what it does is it receives acetylcholine. Acetylcholine will bind, and this allows for sodium to be brought into the cell. And this changes the way our biology. We're not entirely sure how important acetylcholine is, but we know that they play a huge role in some of the brain diseases we've talked about throughout the semester and in our lives, such as Alzheimer's and Parkinson's, etc. Choline ligand-gated ion channels cannot be underestimated in their importance. And as beyond even acetylcholine, you can see that channels and receptors and transporters in general are huge drug targets. So can you take a look at this list and see if you can identify some of the impact of these drugs on these channels? What would happen if we turned off the potassium leak channel? What would happen to the body? Is this a good? What case would we want a drug like this? So I want you to spend some time with that and look at that and see. We'll talk about that more in class, but I expect you to have some ideas about this when we come to class to talk about Chapter 12, because I want you guys to be able to discuss some examples of how drugs interact with these things. And as always, if you have any questions, please let me know.